you. It's a pleasure being here, and thank you to the organizers for giving me the opportunity to come and speak to you today. I'm going to be reaching down a little bit to, so pardon if I'm bouncing up and down with the slide presentation. Basically, we're required at the University of British Columbia to give a disclosure if we have any kind of potential conflicts of interest, and obviously I have one today. I'm the major shareholder of Conexus and its founder, and so we offer services and products, as you'll see through this presentation and later on today in the workshop. And we're supported in part from grants from the National Research Council of Canada and the National Sciences and Engineering Research Council of Canada. Okay, so when we take a look at where we're at in proteomics, we look at genes and they're regulated by methylation. And in this case, if genes are unmethylated and they're activated by transcription factors, we're going to have the production of mRNAs. And we can monitor mRNAs with microarrays. The problem with the mRNA levels is that they're altered by microRNAs, these messenger RNAs, as well as by RNases that degrade them. So they're not necessarily translated into proteins. And the proteins themselves can be undergoing proteolysis and turnover and covalent modifications. And in most cases, when proteins are first made, they're not really active. So to get a sense of the state of activation of a protein kinase, or of proteins, we need to look at when they're fully modified and processed. And if one is trying to understand the phenotype of a model system, really that's a reflection of the amount of active protein. And we don't get that information from mRNA microarrays or looking at the methylation of genes. We have to look at the proteomics level, and that's a much more difficult task than our genomic counterparts have. Because ultimately, if we want to alter phenotype, we need to be able to use drugs to modify that. And so protein kinases in particular are a very fertile area of investigation. As you'll see, about a third of all the R&D going on in the pharmaceutical industry is targeting protein kinases. So really, that's where, to me, the action is. And so protein kinases, they simply catalyze the reaction where so protein kinase catalyze reactions where they phosphorylate proteins with ATP. And one of the things that we've just released in the last couple of weeks is the KinaseNet website. Now, we have what we call the Signet Knowledge Bank, which is composed of several knowledge bases. These are all open access, so everything I'm presenting to you today is free, and it's available for anyone to access it at any time. And so the KinaseNet website has detailed information on about 536 of the known human protein kinases. There are some estimates that the number may be as high as 568, but the information on these other 30 kinases, which are all what we call atypical kinases, is not yet in a consolidated form. So this is a very detailed database on protein kinases. I think it's the most detailed database available in the world. And when you pick the name of a kinase, and you can get there with www.kinasenet.ca, CA because we're based in Vancouver, Canada, and you'll get a listing of all of these different sites. Actually, maybe I should use the pointer. Is it right? Oh, here it is. You'll get a listing of the aliases of the protein and detailed information on isoforms and some information about its interacting partners if it's a multi-subunit protein. You can recover the amino acid sequence, the catalytic domain sequence, the nucleotide sequence from this. And if you scroll down the page, it'll show you what other proteins that it's related to. In this case, we were focused on the ERK1 protein kinase, which our lab discovered. And what kind of domains it has, what kind of covalent modifications that it's subjected to, at which sites, an analysis of its expression levels in various human tissues, and the number of samples where these measurements were made. This is based on mRNA expression levels. Also calculation of this percent amino acid identity and similarity of the gene in different species right down to the yeast and fungi. And then a map of where that protein kinase fits in its regulation by upstream regulators and downstream substrates. Red lines are inhibitory, green lines are activating. All of this is what we call a connections map. I'll come back to this a bit later. But if there's one available, we post it. And then a list of all of the known upstream kinases that target the sites on that protein kinase that regulate it. And there's pop-ups that appear with more detailed information on the effect of that phosphorylation. And then a listing of known substrates, and this list can be quite long. In the case of ERK1, there's over 500 substrates that are listed, and whether or not there's a functional effect of that phosphorylation, the sequence of where it's located, the actual amino acid numbering. And then we take all that information and aligning those sequences, we do a specificity matrix. And what this does is it allows us to determine what is it that that protein kinase is looking for in its recognition. So we have a list here of the 20 possible amino acids at the 15 positions surrounding the phosphorylation site at the 0 position. So this is primarily a serine kinase, so it'll do a little bit of threonine. And it's looking for a proline residue at the what we call the -2 position, 2 amino acids upstream of the phospho site. But the most important one is a proline right after the phosphorylation site. So we do this analysis for all 500, about 500 of the kinases that are typical kinases. So that can be useful for designing substrates for those kinases to measure their enzyme activity. And then we have a listing, and there's more details about this in another Signet knowledge base called DrugKinet. And we list all the known inhibitors that have been reported, the lowest reported IC50, KD, or KI value, with links, direct links to these other websites for detailed information on those drugs. And finally, we have a listing of all the disease associations of that kinase with a strong cancer emphasis, but also other types of diseases where that protein kinase may play a role. So ultimately, our goal is if you can take a kinase and you can link it to a disease, and if you can take a kinase and link it to a drug, then you can use the kinase to bridge a drug to a disease. Okay. So if we look at the proteome today, we're looking at about around 21,000 genes. The number seems to vary still, even though it's been many years since, almost 14 years since the genome was first sequenced. Around 21,000 genes. Of those, about 568 are kinases, and with alternative splicing, there's over 1,000 protein kinase isoforms. Now, these kinases catalyze the phosphorylation, and we now believe of over 1 million sites in the 21,000 proteins. We know by mass spectrometry, there's at least 200,000 sites that have actually been experimentally verified. And this raises the question, much like DNA, where when you look at DNA, you have only about 2.6% of the genome actually encoding genes, whether they're for proteins or tRNA, RNA, messenger RNA, ribosomal RNA, or the microRNAs. And so same goes with phosphorylation. We think that the vast majority of phosphorylation that's happening inside cells is actually inconsequential. It probably is not important. So it really begs the challenge that we need to find those phosphorylation sites that actually are critical, that if they're influenced, we control cell signaling pathways that way. And so a lot of the work that we're trying to do is to define those critical amino acids, phosphorylation site. And of course, this is a reversible. The phosphatome, the complement of phosphatases, about 156, catalyze the reverse reaction. And that's one of the distinctive things about protein phosphorylation is that it's reversible. I think this is especially true for tyrosine phosphorylation. I think although there are serine threonine phosphatases, this is less true probably for serine threonine phosphorylation, as I'll come to. So we have 3 types of amino acids that are modified in particular, although there is other types of histidine phosphorylation and some other amino acids that can be phosphorylated. But primarily serine and threonine and tyrosine, and there's receptor and non-receptor tyrosine kinases that target phosphotyrosine. There's about 90 protein tyrosine kinases. The bulk of the rest are serine threonine kinases. There is some dual specificity kinases, that bad name, that do tyrosine, serine, and threonine phosphorylation as well. But they tend to look like serine threonine protein kinases. So when you look at phosphorylation inside a cell, by weight, tyrosine phosphorylation is quite rare. In a stimulated cell, only about 1 in 2,000 residues are actually phosphorylated. Here, we can see about 90% of the phosphorylation by weight is on serine and about 10% on threonine. But if you look by number of phosphorylation sites, only about 4% of the total number of sites that are sequenced randomly are actually on tyrosine. And we have about 40,000 identified tyrosine phosphorylation sites by mass spec. So if 1 in 25 residues are tyrosine phosphorylated and the rest are serine threonine phosphorylated, then you can take 25 and multiply that by 40,000 and you're up to 1 million. So this is actually quite realistic. Most of it's on serine sites, and there's some threonine sites as well. So this is really high stoichiometry of phosphorylation tends to occur here, but you can see here the stoichiometry of phosphorylation even in a growth factor treated cell where you have growth factors binding to these receptor kinases. The stoichiometry is still quite low at individual sites. So these protein kinases, they're linked to, we now know, over 1,300 human diseases. And that creates great opportunity for coming out with drugs to treat a lot of these disorders. And as I mentioned, about a third of all the R&D is focused on kinases now in the pharmaceutical industry. I predict that within the next 10 years, more than half of the development of drugs will actually be protein kinase focused. Because it's proven to be such a fertile area. There's over 27 drugs that are already on the market, primarily for treatment of oncology applications. There's over 150 that are in clinical trials and more than 500 that are actually in animal trials. And this number is actually continually increasing. So this is a very, very active area of research. When we look at the distribution of the diseases that these kinases are linked to, about a third of them is cancer. And that's where a lot of the drugs, about 80% of the pharmaceutical action is now, is on cancer applications. But you can see the neurobiology targets that actually a large chunk of kinases are actually related to that. And then there's other immunological and cardiovascular diseases. But there's a huge area of investigation available. So having drugs that can target protein kinases will be really useful reagents. Now, this is another knowledge base that we've created called www.drugkinet.ca. And this has a listing of, as it turns out, about 106,000 kinase drug pairs where we know the kinase and we know the drug and we have a Ki or IC50 or KD value and detailed information on those drugs. And this is about 850 compounds that are usually commercially available. And these have been tested against at least 400 kinases. So you get an idea of the specificity of these drugs. Now, we use this information on these pairs, looking at the kinase structures, to come up with in silico predictions of another 200,000 kinase drug pairs, where if you pick this kinase, here's some pre-existing drugs that might actually target it based on similarity in structure. And that's used in part by finding what we call inhibitor-determining residues that are located in the catalytic domain of those kinases. So this is, again, an open access website. You can use it to help you find what might be inhibitors of some of these kinases that you can use in vivo studies. And like I say, through the prediction with the inhibitor determining residues on kinases, we were able to come up with additional pairs. We actually have two different algorithms that we use. One's a decision tree method, another is a linear regression model. And with these methods, we're able to actually not only identify in part what drugs might be affecting those kinases, but identifying what amino acids on those kinases might be involved in the interaction with those drugs. So, we take something like the structure of imatinib here, Gleevec, as it's also known, and our prediction algorithms circled those amino acids that our algorithms predicted that would be surrounding the site where the actual 3D structure with the Gleevec shown here and the amino acid residues are located that are in the vicinity. And you can see we actually are able to predict what those interactions are, at least some of them. Now, with 1 million phosphorylation sites, we need some way to track that. So we have our PhosphoNet website here. And again, this is open access. And this website, it actually has not only 177,000 known phosphorylation sites, but we've also predicted another 790,000 sites trained with our algorithms in these human proteins. And this website, it also collects from the literature functional information on these sites, of which is only about 1% of them is such information available. But we've taken each site, the known and the predicted, and done evolutionary analysis in 20 species. The concept is that if a phosphorylation site is highly conserved in evolution, probably it plays a physiological role in the cell or the organism. And so it's been evolutionary pressure to retain that phosphorylation site. And it also has kind of specificity prediction, and it also identifies related phosphorylation sites in other proteins. So as you go to this site, you just enter the name of the kinase or its UniProt ID number or an alias, and it'll pull up the information on its identity and Go terms, and has direct links to other websites, our own in the Signet Knowledge Bank, but also some other important sites like UniProt and Phosphocyte Plus and String, where it'll go directly, in most cases, to the page. Now, when you get a scroll down on the page, you'll get a listing of all the phosphorylation sites, the sequences, and then these navigation bars. If there is a white button, it means that if you take your mouse over it, you'll actually see information on what the effect of the phosphorylation is, or what kinases are known to phosphorylate the site, or phosphatases to dephosphorylate the site. Later we'll be having linkings to our products here. But then these orange buttons, they take you to additional modules where you can get the evolutionary information, or the kinase prediction, or the phosphorylation site matches for those individual sites, depending on which button that you click. So if you went to PhosphoNet evolution, you would get in the 20 different species, the sequence, the actual phosphorylation site number, and there's a listing of its overall conservation scores. And so this is the MAP kinase phosphorylation site, the TEY sequence, where both those sites, when they're phosphorylated, results in activation of that protein kinase. And you can see that this sequence is very highly conserved throughout evolution. Now, occasionally you'll get a mismatch, maybe by 1 or 2 residues. So in fact, the thale crest, the Arabidopsis, the mustard plant, it has the TEY phosphorylation site too. This is actually one of the most conserved protein kinases in evolution, is MAP kinases. And then we have a calculation of the percent amino acid identity and similarity for each of the sites across the species. And then we look to see which amino acids are the most conserved throughout that to define a consensus site. Now, as it turns out, when you look at phosphorylation of proteins and you look at the evolutionary analysis and how they're retained, the sites that are the most retained turn out often to be in protein kinases themselves. And threonine phosphorylation sites are the most conserved, and they tend to be activatory. In fact, if you look at the most conserved phosphorylation sites, they tend to be activation sites with about an 8-to-1 ratio when you're looking at kinases. So the way our algorithms work is that we define amino acids for each for the peptide substrate or the protein substrate where you have a phosphorylation site. For each position, we define those amino acids that are involved in recognizing that interaction. We call these substrate determining residues. We train this algorithm with about 12,000 known kinase substrate pairs where we knew the protein and we knew the phosphorylation site. And then we did studies with peptide arrays. We identified 8,000 kinase substrate peptide pairs using peptide microarrays we'll talk about later this afternoon. And with that training of over 20,000 kinase substrate pairs and knowing the primary sequence of the protein kinase and defining these substrate determining residues, we can actually take any protein kinase that's a eukaryotic protein kinase and predict basically what its specificity is. And it works pretty good. Here you can see data from aligning the sequences of known substrates of many different protein kinases and what our computer algorithm predicts. And you can see that we get quite a nice match in general. So using that kind of algorithm, we took all 1 million sites and predicted with matrices for each of the 500 eukaryotic kinases which kinases have the highest scores for each site, and the top 10%, or sorry, top 5% are actually listed on the website with their scores. So here you can see that for the ERK1 map kinase, for the threonine site that's the activating site, next to the tyrosine site that's activating, the kinases that our algorithm predicts, and once our algorithm is trained, it only needs the catalytic domain sequence, and doesn't have to be just human, it could be any species. And it comes up with a match, and here you can see the most top predicted kinases with that algorithm is MEK1 and MEK2, which is actually known to be true. How are we doing on time? About 10 minutes? Yes. Okay. So the other thing that's kind of useful is there's a lot of phosphocyte antibodies out there, and often people see cross-reactivities with these antibodies, as you might expect, because if there's 1 million phosphorylation sites, not all of them will be phosphorylated at any given moment, but there's a high probability that there will be many, many proteins that can be phosphorylated, and these phosphocyte antibodies are picking up these sites. So what are those sites? Now you can go to mass spec, but it's kind of nice if you can identify some potential players where your target site can be compared to those out of the 1 million sites that have the closest sequence. So this is actually phosphocyte match looks for what that is. And then you can click on these proteins and you can get more information about each of those proteins in their phosphorylation sites. Well, a lot of this is driven by the fact that many of the protein kinases share a common catalytic domain. Tony Hunter's lab was the first to really identify this. They kind of scooped me when I was in Krebs's lab and published this first. But we have these subdomains, this GXGXXG motif that binds the phosphates in ATP, the AXK motif, subdomain 2, where this lysine residue, if you mutate it to any other residue, like an arginine, you reduce the catalytic activity by 100-fold. So these key amino acids are found, and they define the active site of protein kinases. And there is a region here, subdomain 7, with the DFG sequence. It's critical for activity. And this intervening sequence that then goes to where the subdomain 8 region here is, this APE sequence. And a lot of the activation sites in kinases are located in what we call this activation T-loop. And these are where many of the conserved sites are. So most likely this target of a protein kinase is another protein kinase. So these kinases connect to each other in these very elaborate cascades. And so the interesting thing is that when you look at where these activation sites are, they're in this intervening subdomain 7-8 activation T-loop. And these are stimulatory sites. There are some inhibitory sites in the GXGXXG motif, subdomain 1. When you phosphorylate there, like in the cyclin-dependent kinases, you inhibit the protein kinase. So Shen, a graduate student to my lab, we looked at this in the ERK1 map kinase, and because in interest of time, I'll just tell you the net result is that we found that these residues, here's the APE sequence, what's green here in many different kinases are known to be activating sites. And there are these residues here and here that are usually a threonine in this position and a tyrosine in this position that show up frequently in protein kinases as well in the activation loop. And so what she did was she mutated some of these residues, and to make a long story short, this threonine residue that in the activation loop here, all these are the non-tyrosine kinases, and these are the tyrosine kinases. And these are the residues surrounding where the activation loop would be. And here, for example, is the threonine that's critical for, and the tyrosine critical for activation of ERKs. This is the other threonine here. We see that 71% of all the serine threonine kinases have a threonine at that residue. And if you look at identified phosphorylation sites, again, this shows up in 27% of where it's been confirmed to be phosphorylated. So, interestingly, when you look at the tyrosine here, this is also highly found in the serine threonine protein kinases, but never found in a tyrosine protein kinase. This is one of the ways that we identify what's a tyrosine kinase and what's a serine threonine kinase. So this phosphorylation, this site here, which is missing in tyrosine kinases, is extremely common in serine threonine kinases. And so it turns out that these two sites are key inhibitory sites, and that protein kinases, like G proteins, have an intrinsic GTPase activity. They'll turn themselves off. Well, these kinases, serine threonine kinases, have an auto shut-off mechanism that once they're activated by an upstream kinase at the activation sites, the kinases autophosphorylate and they turn themselves off. And this is one of the reasons why many tyrosine kinases can be oncogenes, but very few serine threonine kinases are oncogenes as a built-in protection. It's hard to mutate these. Well, with phosphorylation sites that are driven with a lot of proline residues, about 20% of all the phosphorylation sites in serine threonine have a proline residue after the phosphorylation site. And this is quite frequent in PES sequences. And so with PES sequences, what we see is that they're rich in these serine threonine sites. And it looks as if when you have some seed phosphorylations, it actually drives further phosphorylation of proteins, so you get hyperphosphorylation. So one of the questions is, what is the role of hyperphosphorylation of proteins? And we think that what these do is they actually open up the structure of a protein, much like when you have SDS, polyacrylamide gel electrophoresis. You have SDS coating the protein, and these negative charges unraveling the structures of proteins. We think the hyperphosphorylation unravels the structures of proteins, reveals sites for proteolysis, and actually drives the degradation of proteins. And this is often mediated by a protein. Yeah, it looks like, sorry, none of my animations are running, but anyways. The 20S proteasome is the major complex for this. So if you have short peptides now that have been cut that are highly charged, and this is the active site, by the way, of the 20S proteasome. It looks like a little barrel-shaped structure, and the center of it is where the active site is. Well, it turns out that if you look at the distribution of amino acid residues, the long chain basic amino acids come out in the middle, and the short chain acidic amino acids are in the middle. But the longer chain acidic amino acids are on the outside, and the shorter chain basic amino acids are on the outside. So what you have is an electrostatic funnel that like a vacuum cleaner, is sucking up all these peptides that are negatively charged and bring them to the active site. So we think that this form of regulation of proteins for degradation is probably as important, if not more important, than ubiquitination of proteins. And there's nothing in the literature about this, by the way. Well, we have, with gene expression data, one of the beauties of gene expression is that, at least with mRNA levels, you can have very good probes, and they're pennies to make, and you can make these arrays overnight, and they've been extensively used. And databases like the National Center for Biotechnology have created this gene expression omnibus with data from probably more than 60,000 studies now, microarrays. But we took the data from that, we normalized it, and we were able to come up with, for the 21,000 genes, averaged values for the expression levels of all the 21,000 proteins in about 600 different types of tissues and cell lines and cancers. And so this TranscriptoNet website has that information, and it's queryable by model system or by the protein. And so it's, again, open access. And it has about 125 million measurements of expression. And you can also use it to identify the most commonly expressed genes, the most uniquely expressed genes for a tissue. You can compare two tissues to see how related they might be. All these are queries that you can do. You can see what genes are the most expressed in human cancers. That's all online. And what genes are highly co-expressed. What we found was that if you look at the data from the string database, which gives you protein-protein interactions with real protein data, and you compare that with the data from mRNA co-expression, there's almost no correlation at all. So just because genes are co-expressed, it doesn't mean that the products of those genes interact with each other. It actually correlates quite poorly. Okay, so we also have a cancer database, AlcoNet, that is accessible now, and it has not only the data on gene expression that we've had from TranscriptoNet, but also has mutation information. And you can query it, unlike, for example, the COSMIC database, where you can know a protein and look at its mutations. Here you can also look at a tissue and find out what are the genes that are most commonly mutated in those tissues. So this can be very useful for defining oncoproteins and tumor suppressed proteins. And unfortunately, I can't talk about it now because I don't have enough time, but tumor requiring proteins, which are third category of cancer proteins that could be targets. So the problem, of course, is that we have all this information coming about the expression of these proteins and their regulation, and it's complex. So how does it all fit together into a signaling pathway? 2 minutes. And we've created a new website. We're about to launch this. But we have two pathway mapping initiatives. One's called Connections Maps, and I'll talk more about that later this afternoon. In this case, we have all these phosphorylation sites, whether they're inhibitory or activating for a target protein like JAK2. We list the upstream kinases that target those sites, and we list downstream substrates and interacting proteins for those sites. And so with these kind of maps, you're able to navigate a little bit for high-density information, I think in a more intuitive kind of fashion than typical maps that you see. Now we have a second type of map, Atlas, and this is going to be customizing interactions between drugs and protein kinases, between proteins and proteins, and also kinases and their substrates into pathways. And these interactions, they have a different kind of a conformation from what you're typically used to seeing. Atlas is still in development. It will be online shortly. And it'll show experimental and predicted interactions. And we have some of these maps already working, for example, for drug-protein interactions. You just put in the name of either the kinase you're interested in or the drug you're interested in, and then generate the map. And then you'll get a map which kind of looks like this. And you can customize these maps online, so you can look just at predicted interactions, which will be dotted lines, or confirmed interactions, which are solid lines. So here is our target track A, the NGF receptor, and these are all the drugs that are known to interact with it. And the closer it is, the stronger the interaction is known to be. And you can follow this and see that a lot of the predicted interactions are matching what we have with our confirmed experimental interactions, showing you the power of this algorithm for prediction of drug interactions. And the nice thing about this is that when you take your mouse over it, it pops up a little menu with links to other websites with detailed information on that drug. And if you click on the drug, it puts it at the center of a new map, and all the kinases that interact with that drug are then featured. So you can hop through signaling systems using this kind of a map. We have this also for protein-protein interactions with detailed links on, here's your target protein, the insulin receptor, and this is actually generated. This is a real image from the analysis, with the proteins being color-coded, and you'll get these links to different pathways, different protein-protein interactions. The more it's been reported in the literature, the closer that those interactions are on the map. Also, the larger the icon, you can customize this to different types of tissues. So if you pick a particular model system, it takes transcriptomet data, and the size of the icon is a reflection of the level of that protein in that tissue type. So you get different maps for different tissues. And then later we'll have our last module we're working on is the kinase substrate interaction module, where you have a target upstream regulators and what sites are phosphorylated, and downstream targets of those kinases. And again, with pop-up menus and hyperlinks to other websites and the transcriptomet website. So finally, all of these interactions are looking at the evolutionary analysis, looking at the strength of these protein kinase substrate interactions. Here we've started to put them together where inhibitory interactions are red and activating ones in green. And this is just a mapping now linking of all the kinases with each other to determine the structure of the human kinome for the critical part. This is kind of like the highway map of the United States, not all the little roads, but where is the main flow of information happening? And so we're developing antibodies to track these proteins to then see, are these the only pathways, or is there in fact more extensive pathways? So I'm sorry that I've already run as a chair a little over time myself, but this is obviously a reflection of the work of a lot of different people to create these websites, but they're there, they're free. We hope that they will be useful in guiding your research. Thank you for your time.
Thank you.